whole career I was funded.
It's amazing.
And then, and now to see this taken off, I mean, if you wanted a seminar about psychedelics back in 1980, the only person who could give it in the world probably was me, or one of the few. And now it's like somebody told me there were 600 organizations now trying to commercialize psychedelics as therapeutics.
That's quite incredible to witness such a development.
Yeah, unpredictable for me.
And kind of it being mainstreamed through a lot of this kind of paradigm of wellness and health, and does that make sense to you, would you say, or is that kind of indicative of maybe how health oriented society tries to be anyway? Or
well, I read a lot of the early literature, and it was replete with anecdotal stories of people who had a high dose experience and witnessed the Big Bang and so forth and so on, and contacted something that seemed like God, and their lives were transformed ever after. And so I always knew they were really powerful, and I had some really powerful experiences of my own. But imagining that they would be used to treat depression and anxiety and addiction and all these other things, there just wasn't any database for that. There were all these psychiatrists who were using it, were really excited about what they were doing, but they weren't controlled studies, and they didn't have good outcome measures and the kind of clinical studies we have today. So it was clear. I mean, imagine all these old societies, the shamans used these, they knew they were powerful. Just today, I heard a lecture by a guy at Purdue. He was a plant pharmacologist, and he said that the people that used these mushrooms were so primitive that they hadn't even learned how to make alcohol. And I thought, that's a pretty stupid comparison.
Yeah, that's very strange. Using alcohol as a sort of metric for intelligence is one thing I've never heard before.
I was sitting in his lecture, and I was floored by that comment. So primitive, they hadn't even learned how to ferment alcohol.
So ridiculous.
So what time do you want to start this? I see it's 2 minutes past the hour here.
Filling up now, so I suppose in the next 2 minutes, is that okay? Or if, otherwise we can just get started now. It's honestly up to you. But yeah, it's interesting thinking about how there is this real emphasis on these kind of things as being like discoveries, or that we've found out that this can help with depression or anxiety, whereas the curative procedure has been established in indigenous cultures for a long time. Mescaline, for example, is phenomenal to see how feels like the West is sort of rediscovering a lot of this stuff rather than
We had the Eleusinian mysteries in ancient Greece that lasted 2,000 years. Every famous Greek you could name probably participated in that ceremony at least once. We don't know anything about it, because to speak about it, they put you to death if you told anybody about it. But one poet said, "Whoever has participated has seen both the beginning and the end of time." So, what does that?
Yeah, exactly. I know, it's interesting reading a bit into like the different speculations of what exactly it could be. Some people talking about it as if it were an ergot derived substance. I don't know how likely that is. Surely, because
I've talked to a lot of people, and I've had conversations with people who had different proposals, and was it a mushroom, was it ergot? Because they really don't know. All they know is it had mint and barley in it. They said, "Oh, barley, it was probably ergotized barley," but ergot is a pretty nasty.
Right.
So I don't know. And the Greeks, when they talk about wine in Rome, it's the same thing. Wine was not the wine we think of. I mean, if you went to a party in Greece or ancient Rome, and people would talk about how you only had to take one glass of wine to knock yourself on your ass, because they would put in henbane and atropine and all kinds of toxic things. And some parties that they had, the wine actually killed people. And they said, oh, the wine was too powerful. A bunch of people died. So, who knows what they had in those, in the Kaikion.
Yeah, I'm personally a bit skeptical of the whole ergoline explanation, because it's like, how on earth could you even do that without causing things like gangrene and death? The process from ergotamine tartrate to LSD is obviously so distant, so I couldn't see how there would be any historical parallel. But yeah, no, it's really interesting. And well, I think we can get started now then, if that sounds good.
Sure.
Great. I'll just give you a quick introduction then. Not that you need one, but this is, we're excited for our second talk, where we're being joined by Professor David Nichols, the American pharmacologist and medicinal chemist. Professor Nichols is one of the world's top experts in psychedelics, having worked in the fields of psychoactive drugs since '69. And having published approximately 250 scientific reports and book chapters. His contributions include the discovery of escaline, LSD, 6-APB, and I could go on forever. Anyway, thank you, Professor Nichols. I'll let you get started on that.
My screen. And this is probably it. Can people see that? Okay.
Yes.
This is going to be kind of a basic overview, some history, and I'll talk about a project that I did and a couple other things that I did that just contributed to the field, just to give people some information about the kind of things I've been involved in. Starting off, psychedelics, this is an official definition out of Goodman and Gilman that was in the 7th and 8th edition. And basically, they said that the definition of a psychedelic that makes them different is the fact that they produce experiences that cannot be, feelings that cannot be experienced otherwise except when you're dreaming and having in religious exaltation. And that's unusual for a class of drugs, as we know. Usually they would say it would lower your blood pressure or lower your anxiety, but to produce an effect like a dream or a religious exaltation, a kind of a religious epiphany, that's pretty amazing. So the publications, this is the number of publications tracked. This came out in a paper in 2021, and we're cut off here at 2020. But you can imagine this is going on up above the, let's see if we can make this up above the 500. But the number of publications, I started graduate school in 1969, which is kind of right here, just before things started to drop off. And not much happened. 93, we had 93, 94, Strassman published his studies on DMT. But then the publications started increasing. 2011, 2016 were key publications on psilocybin use for treating depression and end of life. Then we had one from Robin Card Harris at Imperial College. And so it's really taken off. And I've been tracking, I keep a file of these reprints when they come out. And I have, way over 1,000. I mean, even NBOMe alone, which is not a classic psychedelic, there are 1,900 publications in the scientific literature just on NBOMe. So this has really taken off. The Controlled Substances Act in the United States was passed in 1970. I'm sure there's something in the UK that's comparable. I don't know when it was passed, but the UK led most places. They said, you have to make psychedelics illegal or you won't get any help from us. But anyway, you can see it rose up, dropped off, and now it's taken off, and it's probably up here someplace off the charts. And so the natural psychedelics are the ones that we're most familiar with, and there are 3 classes: phenethylamines, tryptamines, and lysergamides. This is a little baby peyote cactus. This is used in the American Southwest, and it contains a substance known as mescaline. It's a phenyl, ethyl, and ethyl is a 2-carbon piece. Amine. It's a phenethylamine with 3 methoxy groups. It's orally active. It lasts about 8 hours, 8 to 10 hours. The dose is 250, 350 milligrams. Not very potent, but it's classic and is used in the form of peyote by the Native American Church in their worship services. Then tryptamines, several varieties of psilocybe species contain tryptamines. So if we don't have anything attached over here at the 4-5 positions, if there's nothing here, that molecule would be DMT, which is a naturally occurring tryptamine. It's only active if insufflated through the nose or smoked or injected. If you have an oxygen at the 4 position, an OH here would be a psilocin, and if it had a phosphate attached to it, it would be psilocybin. That's the major psychoactive component in the so-called magic mushrooms. And in those cases where we have an O with a phosphate, the body has an enzyme that clips the phosphate off. And psilocin, 4-hydroxy here, is the actual active species. There's one called 5-methoxy-DMT over here that's harvested from Bufo alvarius, which is the cane toad, and it's also only active if smoked. Psilocin or psilocybin is orally active. Lysergamides are ultimately derived from the ergot fungus, and this is an example of rye that's been infected with the ergot fungus. So these little black pieces are sclerotia. They're called ergot, and they are little hard, dark kernels that represent a sort of a hibernating form of the fungus. And they contain ergolines. Ergolines are a 4-ring system. They may or may not have a double bond here, but they have these 4 fused rings, and they may have different things up here. This would be a lysergamide. For example, lysergic acid would be based on this structure. If these R groups were ethyl groups, 2-carbon ethyl groups, that would be the diethyl amide of lysergic acid. So that would be LSD. And those lysergamides are basically fairly potent. Of course, we know the dose of LSD is 1/10 of a milligram, typically, maybe 2 or 3 tenths for people that take high doses, but extremely potent and orally active. This again is a picture of ergot. And so these are the sclerotia, the actual ergots. So if you bought a bottle of ergot, it would just be these black things. And if you put one of these under conditions where it'll sprout, that's actually what this is. This is an enlarged picture of one of those ergot sclerotia. And you can see it's actually producing fruiting bodies. So this is a fungus, but it's a dormant form of the fungus, and it will sprout and grow these fruiting bodies, which have spores, and that's how it propagates itself. In various pastures, claviceps Claviceps paspali on tall fescue grass or on Dallas grass, in fields where sheep and cows graze, if they become infected with these ergot fungi, they can make the animals really sick. In the old days, back to the Middle Ages, there were people that ate bread that was made with ergot contaminated rye and other grains. And these are really toxic alkaloids. The ergot alkaloids present in ergot basically cause a constriction of your peripheral blood vessels. So in your fingers or in your toes, they'll produce prolonged constriction. And people would continue to eat bread because it was basically a staple of their diet. So if the bread was contaminated with ergot, eating it continuously, what you get is ergotism. So these examples at the bottom with the fingers and toes, this is what ergotism looks like when it's really extreme. You get gangrene. The blood vessels stay constricted so long that it causes necrosis and death. And so people that would survive this would be missing fingers and missing toes. And this is sometimes called St. Anthony's fire because it intensely burned and it produced an intense burning sensation in the fingers and toes. And these people would also suffer hallucinations. This is ergotamine, which is one of the alkaloids that's in the ergot fungus. That's a vasoconstrictor. And you can see it's got this 4-ring ergoline system here at the bottom. And whereas LSD has a diethyl amide, we have this complex tricyclic system up here attached. So the ergot alkaloids in general have more complex pieces of organic shrubbery up here. LSD is a fairly simple molecule with just the diethyl groups, but this is another example of an ergot alkaloid. Ergine or lysergamide, where we only have an NH2 here, is again an ergot with again, it's a primary amine that occurs, it grows in seeds of ololuki. This is a morning glory that grew in South America. And these seeds were ground up and made into an infusion when they couldn't get the magic mushrooms. And also Hawaiian baby wood rose is a member of this family. It also has lysergamide or urgine that grows in the seeds. The plant doesn't actually produce it. The seeds become infected with an ergot fungus that's symbiotic. And so it's produced by this fungus living in the seeds of morning glories or baby wood rose. In addition, we probably all heard about ayahuasca, which is a tea made from a combination of two things. Psychotria viridis or chacruna produces DMT in the leaves. And I showed the picture of a DMT in the earlier slide. DMT is not orally active because if you take it orally, it's broken down by monoamine oxidases in the liver, so it never gets into your system. However, this is Banisteriopsis caapi, which is a big vine that also grows, and it produces alkaloids called beta-carbolines. And beta-carbolines inhibit monoamine oxidase in the liver. So if you bust up this, the bark from Banisteriopsis, that's the red pieces here, they break it up in pieces, mix it with the leaves from Psychotria here, and then just boil the dickens out of it. You get a brown, kind of a mucky thing. You filter out the plant material, and that's ayahuasca. So it's got a mixture of DMT and beta-carbolines. These block the enzymes that would normally break the DMT down. So it makes it essentially an orally active form of DMT. Now, when Albert Hofmann synthesized LSD in 1938, Albert Hofmann was a really talented natural products chemist. He finished his PhD in a couple of months and went in to talk to his professor and said, "What do I do now?" He said, "Yeah, write up your thesis." But he said, "I've only worked a couple of months." And he says, "No, you solved the problem." He went to work for Sandoz Laboratories, and he worked on the first project. It was some squib alkaloids for cardiac effects. But he'd heard about the ergots, and he knew they were really potent. So he convinced management to let him work on the ergot alkaloids. And they cautioned him. His research director said, "You know, these things are expensive. They're hard to get. So you be really careful on it." But because Albert was such a good chemist, they let him work on them. And he made a whole series of ergot alkaloids, some of which got into medical practice. But there was a compound called chloramine or nicotinamide, shown here on the right, and it was a respiratory stimulant. And he knew that the ergot alkaloids were extremely potent. Chloramine is not very potent. So he thought if he took chloramine, this molecule, and put it on top of the ergot system, where I've got it highlighted in red here, that he might get a really potent respiratory stimulant. So that was the rationale for making it. And he didn't discover its activity until 5 years later in 1943, because when he sent the LSD down to the pharmacology lab, they tested it in mice. And mice got a little bit excited, but pharmacology said, "This isn't anything we're interested in," so they forgot about it. And he was eating his lunch in 1943, and he thought, "You know, I just have a feeling about this LSD." It was LSD 25. It was the 25th in a series of compounds he'd made based on ergot. He just had this feeling, was translated as a peculiar presentiment, an intuition, that the pharmacology people had missed something when they had screened this LSD molecule in the very beginning. And in industry, pharmaceutical industry, that's pretty atypical. If I was working in a drug company and I made a bunch of compounds and they tested them, they told me they weren't interesting. If I went back and I says, "I think you guys, we just have birds trying to fly through the house, and every time one hits this window, it makes me sad." Anyway, so if you took it down and told the pharmacologist, "I think you screwed something up. This compound is really interesting. You just must have missed something," they would just probably run you out of their lab and say, "What are you talking about? We do good work. Don't come in and bother us." But he didn't get a chance to do that because he somehow got some in his body on a Friday afternoon, April the 16th. And he didn't know how. I've talked to Albert. He didn't know how he got in his body. He was a meticulous chemist. But back in those days in 1943, they didn't wear rubber gloves in the lab, which they do now. So somehow some of the stuff got in through his skin, and he got intoxicated, went home, and thought about it over the weekend and thought, what was I working with that could have done that? And he finally decided it must be LSD. So he made a fresh sample, and Monday went in and took 250 micrograms, a quarter of a milligram, because he thought that was a dose that would be so low that it wouldn't really have any effect. And if you know anything about the dose of LSD, 250 micrograms would, kind of knock you on your butt if you weren't expecting it. So he thought he was dying. He rode his bicycle home. His lab assistant followed him home. They called for the doctor because he thought he was dying. The doctor came and checked him out. He was fine. But that was the beginning of the knowledge about LSD. Sandoz then made it. It was marketed under the name D-Lysid, and they sent it or distributed it to almost anybody that wanted it. You could just write to Sandoz in 1945 or 46 and say, hey, can you send me 10 ampules of your LSD solution? And they would send it to you. It was back before the days of the drug wars and all that, so there was no paranoia. So psychologists were taking it and giving it to their patients, and they thought this was really a wonder drug. So the interesting thing about LSD is it also informed us about what was going on with the neurotransmitter called serotonin in the brain. In 1943, when Albert Hofmann discovered the effects of LSD, there was no knowledge of brain chemistry being involved in mental illness. It was all based on Freudian psychodynamics. And so if you had a child that had suffered from schizophrenia, the story was that you had not been a nurturing parent, especially mothers who were the ones that mostly stayed home. They had a term, refrigerator mothers, for cold mothers who just were distant and not, nurtured their children. So it was all based on the parents' fault. It was the parents' fault. So in about the same time, 1943, they were starting to work on isolating serotonin. It was found in blood clots. And so in 1953, 10 years later, they discovered serotonin in the brain. A woman named Betty Twerog, working at the Cleveland Clinic, had developed an assay for serotonin in animal tissues. And she was looking at blood vessels and heart and so forth. And they knew that most of the serotonin in the body was in the heart and in the intestines, and there was no recognition there was any in the brain. And she went to Irwin Page, the clinic director, and said, do you mind if I analyze some brain tissue? And he laughed at her and said, everybody knows there's no serotonin in the brain. Well, she got some cow brains and rat brains and looked at them, and sure enough found serotonin. And the similarity between the structure of serotonin and LSD made people start thinking maybe there was something about this serotonin in the brain that was causing, mental illness of some kind. So there was a hypothesis just a year later that it was LSD was blocking receptors for serotonin. And about a year after that, they changed the hypothesis to say LSD was stimulating receptors for serotonin in the brain. And if you look at the scientific literature, up until about 1954, everything you read about serotonin is that it was involved in blood clotting and maintaining vascular tone in the blood vessels, etc. And it was in the gut. And then you start seeing pictures showing, publications showing up saying, oh, it's in the brain, what's it doing in the brain? And this is almost contemporaneous with the introduction of chlorpromazine for treating schizophrenia, because that also was one of the drugs that started changing perceptions about biological psychiatry, because psychiatry up until about the early 50s had all been focused on psychodynamic theory and Freudian and Jungian theories. This is a quick dose and route of the compounds. Mescaline, 200 to 400 milligrams orally, 8 to 10 hours. LSD, 0.1 to 0.3 milligrams of the tartrate orally, 8 to 10 hours. Psilocybin, 10 to 30 milligrams orally, 4 to 6 hours. DMT smoked, 30 to 100 milligrams. 5-methoxy-DMT, 10 to 15 milligrams insufflated or smoked, 15 to 20 minutes. This is probably the shortest acting 5-methoxy-DMT and maybe one of the most powerful psychedelics that are known. This is what also occurs in the toad venom. And various phenethylamines largely made in Sasha Shogun's lab, a bunch of which I also made, and with doses ranging from 2 to 50 milligrams with variable durations of action. And the effects depend on your mental expectations, and setting. People think that Timothy Leary was the first one to define these, but actually it was Captain Al Hubbard, Captain Trips, who first told people about set and setting. It goes back before Leary. But anyway, I used to use an example when I taught pharmacy students. I taught a lecture on drugs of abuse and psychedelics. And I said, if you take LSD and you read a bunch of books about Eastern philosophy and meditation and things like that, and you go to your church or synagogue and listen to evocative music, and you expect maybe to have religious conversion of some kind, the probability is high that that's the type of experience you'll have. If you take the same dose and go to Freddy Krueger or Friday the 13th movie, some horror flick, you won't have a religious experience. So this is just set and setting. What your expectation is and where you take it really determines the outcome. And that's unique probably to psychedelics more than any other type of drug. So what are the receptors that psychedelics act at? The major targets for the phenethylamines like mescaline are the serotonin 2A and 2C receptors. For tryptamines like DMT, psilocin, psilocybin, so 2A, serotonin 2A receptor, the serotonin 2C receptor, and the serotonin 1A receptor. LSD again hits the serotonin 2A, 2C, and 1A receptors, and then is fairly promiscuous. It has activity at a lot of other G protein coupled receptors. A lot of experiments have been done that really focus on this serotonin 2A receptor as being the target. It's what they all have in common, and it's where they have high efficacy, high ability to interact. So where is that receptor? Well, if we do a cross-section of a human brain, a slice like this, and we incubate it with a radioactive molecule that binds to serotonin 2A receptors, in this case, it's tritium-labeled ketanserin. The high levels of binding are red, low levels are blue, and you can see that you have these high levels of ketanserin binding in the outer layers all the way around in the cortex, even back in the visual cortex, parietal, frontal, prefrontal cortex. And if you blow that up and use microscopy, what you look in and see is that the ketanserin is binding to these triangular shaped pyramidal cells. And these are the computational cells that are densely packed primarily in the cortex. And so the serotonin 2A receptors are expressed on the apical dendrites, the part of this neuron that goes into the cell body. And you can see it's darkly stained here. So those are pyramidal cells in the cortex, and they're the major computational units in the cortex. This is a layer 5, and they're in layer 5 of the cortex. If you look at the cortex, this is a mouse somatosensory cortex. The outer layers of the cortex out toward the skull and the dura, layer 1, 2, 3, 4, 5. And layer 5 apical dendrites, here are the pyramidal cells down here at the bottom, these big red things. Layer 5 apical dendrites are in here, and those are excitatory. The red fibers are excitatory, the blue are inhibitory. So these are like the central processing units of a computer. These pyramidal cells, and it's like a parallel processor. If you know anything about computers, you have parallel processors. So the cortex is like a giant parallel processor. And these pyramidal cells are like the CPUs, the central processing units. So they take information that comes from all the other areas of the brain, the information about the things we hear, the things we see, feeling, imagination, memory, it all impinges in. And these cortical cells, pyramidal cells, really kind of determine what actually gets acted on to a large extent. So it's not too surprising that if you muck with those serotonin 2A receptors, you're going to change the way the brain processes information. And when you activate those serotonin 2A receptors, it depolarizes the membrane potentials of these apical dendrites here. So it changes their electrical properties, makes them easier to fire, increases the gain, and basically changes the way your brain processes sensory information. So what happens inside the brain cell when you activate these receptors? They're embedded in the neuron membrane. So this is an example of a G protein coupled receptor. This is actually a beta receptor. And what I've shown in gray here, this is the inactive receptor. And what I've shown in pink is the active receptor. After a ligand binds up here in the top region of the receptor, there's a binding site, what's called an orthostatic binding site, down here in between helixes 3, 4, 5, and 6, down inside here. It binds and it changes the shape. You can see the way this is moved. So the pink one is the active one. And what happens is there are loops. This receptor is a long protein that threads in and out of the neuronal membrane like a snake. Sometimes they're called snake plots. So it'll go in, out, in, out, in, out. And there are 7 of these what are called transmembrane helices. So this is helix 5, helix 6. And so when they change their shape, these loops that connect the bottoms of the helix have to change too to allow for movement of the helices. And when they change the signaling components that are inside the cell. So here's an actual to scale picture of a receptor, and it's connected to this G alpha protein subunit. They're heterotrimeric G proteins. So heterotrimeric, so it's trimers. That's G alpha and G beta and G gamma. They're all associated, and they bind GDP, and they couple to the interior of this receptor, which has these loops connecting the alpha helices of the receptor. So when something binds up here in the receptor and changes the shape of the receptor, it changes the shapes of the loops that are down here that interact with the G alpha subunit. That causes a dissociation of the G alpha subunit, and it dissociates from the gamma and beta and gamma subunits, and they all go to other enzymes inside the neuron and stimulate signals. So they might increase the level of cyclic AMP, or they might change a potassium channel and allow more potassium to go in or out. So there's a number of signals that can occur, but this is really the business end. Sometimes you'll see in a textbook, they'll have a great big picture of a receptor and pictures of these little G alpha, beta, gamma, heterotrimeric G proteins, but this is to scale they are. The receptor is just a little switch that basically activates the G proteins, which are the business end of the receptor. And again, this is the membrane of the neuron. And so this is inside the neuron, affecting the biochemistry of the neuron. Just a simplified example. So here's an example, 7 transmembrane helices. And when it's activated by something binding up here, it changes the loops inside, and it can couple to different types of heterotrimeric G proteins. So we have G alpha I, G alpha Q, G alpha S. There are 14 different kinds of G proteins. It can also be acted upon by a G protein receptor kinase. A kinase puts phosphate groups onto certain amino acid residues. So this kinase can phosphorylate serines and threonines that are on this C-terminal part of the receptor. And when it gets phosphorylated, it recruits another signaling molecule called arrestin. Arrestin can cause the receptor to be internalized, and LSD and psychedelics will do that. So if it's highly activated, if this is LSD, it will recruit arrestin, and arrestin will pull it into the cell membrane, so it's not available for interaction anymore. It's called internalization or desensitization. And also there's a whole MAP kinase signaling pathway that even goes into the nucleus that regulates transcription. So depending on what the ligand is, it'll activate different signaling molecules. So this is called functional selectivity or ligand-directed bias. So these receptors, they're not static. They're not just sitting in the membrane. They're always kind of wiggling and shaking around, and they'll go to more stable states or less stable states. And so there are a number of more stable states that they tend to go into. And those more stable states will bind specific ligands. So serotonin, imagine serotonin gets in here. It's a small flexible molecule. It binds to a certain state of this receptor and will activate certain signaling pathways. LSD is a big, stiff, sturdy molecule with 4 rings hooked together. It gets inside there and it changes the shape completely. So you get different kinds of signals produced from LSD. Other types of things like tryptamine, psilocin, for example, might produce different amounts of these signaling signals. So it may activate G alpha Q. There are a whole bunch of these I'll show you on a slide. So you get a whole mixture of signals, and we don't really know which of these are most important for the actual psychedelic effect, but that's a problem that's going to be solved within 5 to 10 years. So here's an example that came out of the lab where I'm working. They developed a tool called TruePath. And what you do is you can get cells and you can specifically infect them with one type of G protein. So here we have G alpha I1, G alpha I2, G alpha I3, etc. Here G alpha Q, these are the ones that the psychedelics activate. G alpha Q, G alpha 11, and G alpha 15. Now the black curves here are just a standard curve of neurokinin, just to show that the system is working. And so they don't mean anything. The important thing is we have serotonin as a red curve, LSD is an orange one. Here's an N-benzyl compound as a dark blue one. And you can see all of these are producing signals, G-alpha Q, G-alpha 11, and G-alpha 15. But in none of these others, they're all flat. So this is a kind of study you would do to determine the relative specificity of a molecule that interacts with the serotonin 2A receptor by seeing which of these pathways were lit up. This is a work in progress. The lab where I work is doing a lot of this kind of thing right now to really figure out how these things work. So when I was at Purdue, how did I assay these compounds? I always had students who were willing to take them, but of course I couldn't do that. So what did I have to do? If you want to understand what these do to study consciousness, really you need agents that perturb consciousness. And the guy who did a lot of that was a guy named Sasha Shogin. If you know this field, you've heard of Alexander Shogin. This is actually a picture of what he looked like. And he made these compounds in his lab. He took low doses and then took increasingly higher doses to see if they had any effect. And if he got to an effect that he enjoyed, he shared them with his wife, Anne, who's still living. She would take them, and if she thought it was an interesting molecule, he had a whole group of professionals who live in the San Francisco Bay Area who they would get together and have kind of a party, I guess, where he would weigh out samples for everybody and they would take them and they would write down their experience. And he wrote two books. One is called PIHKAL, P-I-H-K-A-L, which stands for Phenethylamines I Have Known and Loved. And then he wrote one called TIHKAL, T-I-H-K-A-L, which is Tryptamines I Have Known and Loved. And if you've seen those books, the first half of PIHKAL is autobiographical, but the second half are the chemical recipes for making all the compounds that they took, as well as write-ups on what the doses were and how the experiences were. And in the UK, they used that book to decide how to schedule all the drugs. They went through and they took every drug that he had in that book, whether it was active or not, and they're all scheduled. So anyway, I couldn't do that. So I had to punt and use rats. And you can teach rats the effects of a drug. If you give a rat LSD, he will learn to recognize the effect of that LSD or mescaline or whatever. So I used a two-lever drug discrimination procedure. I've taken away two sides so you can see, and there are these two levers I can press on. And there's a food pellet delivery system here that gave him a 50 milligram sucrose pellet, which is basically a rat candy. And you can train these rats over a period of 2 to 3 months so that if you give them LSD, they'll always press, for example, the right lever. If you give them just a placebo, just a saline injection, they'll always press the left lever. And you reverse. We had half the rats trained with the drug on the right, half the rats trained with the drug on the left, just so we didn't get any positional preference. And so they will very reliably press this. In fact, press it thousands of times in 15 minutes. And every time they press, they could conceivably get a food pellet. We start off giving them one pellet per one press. And these rats go nuts when they go in there. And they would press this lever like 1,500 times, 2,000 times in 15 minutes. And those pellets, like all scientific equipment, were a little expensive. So we had to restrict their ability to earn so many pellets. So we forced them to press the appropriate lever 50 times before you get one pellet. So then we could afford to do the studies. But anyway, if this rat had been trained to press the right lever on LSD, then we would make an analog of LSD, and we would wonder, does it have LSD-like effects? So we would inject it to these rats that were trained on LSD, and then if he pressed on the right lever, he's saying, I think you gave me LSD. So we had rats trained with amphetamine, we had rats trained with mescaline, we had rats trained with DOM, a lot of different rats. So any drug that's centrally active can be used in this assay. And these rats this is a very sensitive assay. So we used a training dose in rats of 80 micrograms per kilo of body weight. That sounds like a lot, but in rodents, if a rat weighs 250 grams, we'd be giving him about 20 micrograms. That seems like a lot, but it's not a lot to produce in any other behavior. So if we injected a rat with 80 micrograms of LSD and just put him out on the table, he would look completely normal. So you don't get any kind of behavioral. but they can pick up that drug cue, and it's very sensitive. So when I started in 1969, the basic question I asked my research was, well, we knew about the phenethylamines, like mescaline, and I made a bunch of these, and we knew about the structure of LSD, and people drawing pictures saying, oh, well, LSD, you can see that's got this phenethylamine. This is the stereochemistry of active LSD. We later found this was a stereochemistry of active phenethylamines. So superficially, you look at it and you go, oh, man, that's what it is. It's this piece in here. It's really not that piece at all. We spent a lot of time proving that it wasn't, and that they bind completely differently, other than the fact that there is a place in the receptor that interacts with the basic nitrogen. But other than that, this is not a good analogy. But that's what I started off doing as a graduate student, trying to prove one way or another, test the hypothesis that was the way they bound. So I'm just going to talk about one short project that I actually did that was kind of interesting, that we were able to validate almost 20 years later. So anybody that knows anything about LSD chemistry knows that the diethylamide, which is this piece over here, the two ethyl groups attached to the nitrogen, that's absolutely critical for the activity of LSD. If you change that to anything else, the activity drops off by about an order of magnitude. So changing these into methyl groups, single carbon atoms, or changing one into a methyl and adding another one over here, a methyl propyl, or changing it into a ring, 5-membered ring, anything you do, the activity goes way off. So these are drug discrimination ED50s. This is a dose in nanomoles per kilogram of body weight in a rat, where 50% of the rats chose the LSD lever. So this is basically the benchmark for having LSD equivalent activity, 28 nanomoles per kilogram. So we thought, we had a hypothesis that maybe there was a specific region of the receptor where this diethyl group's bound. And so we wanted to probe that and see what was up there. So we made a compound, a secondary butyl. So this has also, just like LSD has 2 carbons and 2 more is 4, this also has 4 carbons. So we kept the same hydrophobicity, or 4 carbons, 1, 2, 3, 4. But this is because they have 4 different things attached, it has a stereochemistry. So this is the R configuration, and if we reverse it, it's the S configuration. So we still have 4 carbon units, but we changed the stereochemistry from R to S. And when it was R, you can see the drug discrimination ED50 was 33, about the same as 28. But we changed it to S, it's 124. So there's something up here that's very sensitive to the nature of that substituent. If you hook the ends of these 2 ethyl groups together and make a 5-membered prolidine, it jumps down even further to 168, much less potent. So it clearly was telling us there was something up here that was interacting with the amide substituents, and it made a difference. And so the diethyl was unique, and Albert Hofmann made 80 or 100 of these compounds, and LSD 25 was the only one that had high activity. So we made an early model of the receptor when they published the first structure of rhodopsin in about 2000. We made a homology model, and we virtually docked LSD. So this is LSD in purple, this is the diethyl groups, and there's a loop. It's called extracellular loop 2 that lays over the top of the binding pocket for LSD. And there's a big residue, a leucine 229 there, that looked like it was close enough to interact. So this is a crude representation, but we thought maybe there's something in the extracellular loop 2 that's interacting with this diethyl amide. So how could we test that? So we thought what we do is rigidify the diethyl amide. So again, here's LSD, and these arrows are meant to indicate that these groups can just rotate around. They're flexible. But what if we tied this carbon, what if we tied it back to this carbon and made a 4-membered ring like this? Well, if we do that, this is called an azetidine. And those methyl groups attached to the azetidine, you can see how they incorporate the 2 ethyl groups. Here's one and here's the other. They can be in 3 different orientations. The methyl groups can be on the same side of the ring. They can be on opposite sides of the ring this way, or opposite sides of the ring this way. Those are 3 different stereoisomers. So we have 2 of them. The ones that are trans are designated SS and RR. So if we look at LSD as a flexible molecule, Here, the front ethyl is projected down, the back ethyl is projected up. We flip them, the front ethyl is projected up, the back ethyl is projected down. And you can see the SS azetidine resembles this one in that the front one's down, the back one's up. And the RR resembles this one in that the front one's up and the back one's down. And if we look at it from the top, here again, we see here's the four-membered rigid azetidine, and you can see this is LSD in one of its conformations in one orientation of the diethyl groups. And here's the other one, and you can see the similarity. So we tested these in drug discrimination, and what happens? Only the one with the SS configuration was active. And this is just to show you in drug discrimination in separate experiments, the LSD was 45 nanomoles per kilogram. The SS was actually a little more potent at 25. And if we went to the cis, where they're both on the same side of the ring, it drops off quite a bit, about 4 or 5-fold. Go to the RR trans, it's quite a bit less potent than the SS. And if we look at their ability to stimulate phosphoinositide turnover through the receptor, you see here's LSD with an EC50 of 15 nanomolar, 23% of maximal. The SS at 19, about the same, 43% of maximal. And then it drops off with either the cis or the RR. So in biochemistry as well as in behavior, we're seeing that the SS trans molecule is the most potent. So it represents the orientation of those two flexible ethyl groups of LSD. So in 2017, I retired from Purdue in 2012. In 2017, I'm working in a lab with Brian Roth, and they determined the crystal structure of LSD in the serotonin 2B receptor. The receptor is similar to the 2A receptor, which is the actual target. So similar enough that it gave us some clues about what was happening. So now here's an actual space-filling picture of that crystal structure. Here's that extracellular loop 2. Here's that leucine 229 I talked about. Here's LSD down in here. So what happens is this loop folds over, it twists leucine 229 down inside, and it wedges against LSD, and there's another tryptophan. It locks it in the receptor. So there is a basis for this interaction. So this is actually the crystal structure of LSD and the serotonin 2B receptor. And now I'm going to show you the SS azetidide, the four-membered rigid analog, which had the LSD-like activity. Superimposed on it, it's blue. And what you can see is if this was the RR, this would be pointed up here, and this would be pointed down here. But you can see this is a pretty good superposition on top of the actual conformation of LSD when it's bound in the receptor. So we had predicted this we had predicted this was a conformation of LSD when it bound to its receptor in 2002. And then in 2017, we confirmed that we had done a correct prediction. So that was the kind of thing that I used to do when I was doing medicinal chemistry. Now, the receptor pharmacology, if you look at those azetidides, there are two signals. The G protein, which would lead to increased calcium, intracellular calcium, or arrestin. And I indicated that arrestin was another signaling molecule. So LSD is purple. The SS azetidide, which is equal to LSD in potency, is green. And the RR is red. And so there's no difference between any of them in looking at G protein coupling. But when you look at arrestin recruitment, you see that the LSD and the SS are almost superimposable, but the RR is shifted to the right. There's a lot of interest now in arrestin recruitment as being involved in the action of psychedelics. So this would be somewhat consistent with arrestin being an important signal in the action of psychedelics. So that leucine 229 in extracellular loop 2 is the one that seemed to be folded over, and we'd seen it was locking LSD into the receptor. And just to show you, this is the size of a leucine. Here's the oxygen and nitrogen. And this is the size of a leucine. This is the size of an alanine. I should have turned that phone off. Just give me a second. Sorry about that. So if we replace this leucine with a smaller alanine, what effect will that have? If the leucine is actually clogging up the receptor entrance so LSD can't get in or out, what if we put a smaller one in? And this is exactly what happens. So if you take the wild type receptor that's got the leucine, and you put tritiated, that is radioactive LSD in, and you let it sit until the receptor has reached equilibrium and it's saturated with LSD. You wash off the buffer, and now you put in fresh buffer with no radioactive LSD, and you look at the rate that the radioactive LSD escapes from the receptor, you get a curve that looks like this. And it takes 6 to 8 hours for the LSD to come back out of the receptor. Now, if you mutate that leucine to an alanine, which we showed was interacting with the diethylamide, and you do it, and now when you do the same experiment, you see the LSD comes out really fast, and within about 2 to 3 hours, the LSD is out of the receptor. If we look at calcium flux, here's the wild type receptor and the mutant where we've mutated the leucine to an alanine. There's no difference in the two. So the signaling doesn't depend upon the LSD being locked in there. But if we look at the ability of the receptor to recruit arrestin, here's the wild type with the leucine, and here's the EL2 where we've replaced the leucine with an alanine, and you see the wild type, we're locking LSD in, we're getting a lot of arrestin recruitment. We put the alanine mutant in, and now it comes off, it comes out, you don't get the amount of arrestin recruitment. There's one other thing. In 2020, we published the crystal structure of LSD in the serotonin 2A receptor, which is the target that we're most interested in. And the main difference there is when we look at the serotonin 2B receptor, there's an alanine here. In the 2A receptor, there's a serine. It's got an OH there. And LSD, the NH of LSD, hydrogen bonds to the oxygen of that serine. And that's not there in the 2B receptor. So here's a case, again, looking at LSD dissociation rate. Here's the wild type, and you see it takes a while to come out. But if we mutate this to an alanine, get rid of this hydrogen bonding component, it comes out much more quickly. So this, again, was validating the orientation of LSD in the 2A receptor. And this is EL2. It does the same thing in the 2A receptor that it does in the 2B receptor. So did any of my work turn into anything practical? Well, I started the Hefter Research Institute in 1993, and we funded $10 million to support, it's basically seed money. We supported the clinical studies of Charlie Grove at UCLA, Johns Hopkins, Roland Griffiths, a study of psilocybin in cancer patients, New York University's study of psilocybin in cancer patients. So we really started funding clinical work because no one was doing clinical work. The National Institutes of Health here won't fund psychedelic work. They funded, I think, one proposal now last year, but they have not been interested in funding this work because these were drugs of abuse. They were worthless, so it didn't have any value, right? So we started getting philanthropists to give us money to support this. And we've supported $10 million worth of fellowships and grants for people to really get this field started again. And kind of some of the things we did, this was Charlie Grove's study published in 2011, Roland Griffiths at Johns Hopkins 2016, Stephen Ross 2016, cigarette smoking, Matt Johnson at Johns Hopkins in 2014. He's doing a much bigger study now. Mike Bogenschutz studied psilocybin in alcohol use disorder, published in 2015. We supported that. He's just done a big study now with 96 patients. And a lot of these other publications, if you go to the Hefter website, it lists all the publications. We supported a Zurich lab, Franz Vollenweider. He has the Hefter Zurich Research Center, has actually a center where he was doing clinical work, mostly with psilocybin for all this time. So he's published a lot of papers that the Hefter Institute has funded. And then recently we had a psilocybin versus citalopram. This was done by Robin Carhart Harris at Imperial College, where they compared psilocybin, 2 25 milligram doses 3 weeks apart versus an SSRI, escitalopram, 10 milligrams daily for 6 weeks. And psilocybin was just the same, just as effective as escitalopram. 2 doses, and actually the first dose, probably the second dose didn't add anything. And he mentions that they just used the SQUIDS as a depression readout, but they had a whole bunch of other endpoints that they measured. And the way you set up a clinical trial for FDA is you decide what the endpoint is, and that's what you tell FDA. But if you look at all the other endpoints, quality of life and all kinds of things, psilocybin was far superior to continuous escitalopram. So if this can treat all these different things, depression, anxiety, and addictions, what's the mechanism? We don't really know, but this is the way the brain normally operates. This would be, you can see this is milliseconds, so it's not quite real time, it's slowed down quite a bit. But your brain is constantly sending these signals through networks, organized networks in your brain. And this is from a paper out of Carl Harris' group. Petri and I was the first author. There's brain connectivity, the way those electrical fields communicate. There are networks, resting state networks. And normally your brain you don't have a lot of things connected. There are local hubs in your hippocampus or in amygdala. There are local hubs where there's a lot of activity, network communication going on, but there isn't a lot of outreach to other networks to connect with other networks. But under psilocybin, the network connectivity expands. And Carhart Harris has called this entropic brain activity. You get dramatically increased brain connectivity where all these different hubs and nodes are communicating with each other. And then when the drug wears back off, it collapses back into its normal placebo or normal resting state. And we think that there's some kind of a reset that occurs here. And that's what everybody's focusing on now. All the labs are now starting to do research in this field, trying to understand what is it that psychedelics do to reset these networks and put them back into a stable, healthy network that's not depressed or is not addicted or whatever. So I think we're looking at a new paradigm in psychiatry. SSRIs have a lot of side effects. They don't work for everybody. Psilocybin won't work for everybody either, but they work with 1 or 2 doses. And treatment of addictions, alcohol and tobacco addictions. We have studies underway now for obsessive compulsive disorder, for eating disorders. So it may turn out that psilocybin really changes the face of psychiatry. And this is all just happening during our lifetime. So it's really pretty amazing. And something I never expected to see in my lifetime. A woman asked me years ago, what do you expect is going to happen with your research? And I said, someday, probably long after I'm dead, you'll be having some midlife crisis and your doctor will send you down to talk to this psychiatrist / shaman, and he'll give you a psychedelic session and you'll get a new perspective and get your mental health back. She says, oh my God, you think you'll be dead by then? I said, yeah, probably, but if the vector's pointing in the right direction, that'll be okay. So I think it's pointing in the right direction, and it's going a lot faster than I ever imagined it would go, from starting off being the only one of the few people in this whole world working on these things and everybody telling me I was crazy, to now, the field is just blooming. So with that, thank you for your attention. I hope you could follow most of this. And of course, I didn't do all the lab work. Most of my graduate students and postdocs did it, and I had an endowed professorship for a while. So I should acknowledge that a lot of people contributed to this story. And of course, my story is a lot bigger than I could ever present in an hour here, but I hope you enjoyed it. Thank you.
Thank you very much, Professor Nichols. That was just fantastic. It was really interesting having a proper insight into all of that. So yeah, is there possibility for any questions briefly, do you think? Or it's fine if not, because I remember this is the time we agreed to end up.
We have time for questions. I don't have another meeting for half an hour.
Okay, great. Brian, would you like to provide a question?
Yeah, I'll just give my quick one instead of my long-winded one. Is there anything about psilocybin pharmacology which gives it preference in psychiatry over other psychedelics like LSD, or is it just a historical reason for why it's used over the others?
The Hefner Institute really started the use of psilocybin I worked on the synthesis. In fact, I made the psilocybin for Johns Hopkins. I made the DMT for Rick Strassman. I made the MDMA for Rick Doblin. When Charlie Grobe he started off working with MAPS, and they wanted to do an end-of-life study with MDMA. And Rick he was on the Hefter board, one of the original founders. He met with it and he said, I don't feel comfortable giving MDMA to dying patients because they're already really sick. And MDMA is a psychostimulant that raises blood pressure, et cetera. And he says, but I want to do this study in dying people. What could we use? And so we discussed, like, what could we use? Well, LSD had been used for terminal cancer patients back in the 60s. But then you're talking about a very profound experience that's going to last 8 to 10 hours minimum. If you're going to do it clinically, you're going to have to keep them overnight. And these studies are not inexpensive. When we did a study at New York University, we calculated that was $25,000 per subject. So Charlie Grobe had a small number of subjects. We didn't have much money at that time. So we said, that's going to be too expensive. It's going to take too long. And we thought if we had a study, and we published back then in 2011, he started a study in 2009. If we said we were giving LSD to dying people, we thought the press would be all over us. Like, why you're giving LSD to dying people? And back in those not that long ago, but LSD was still considered to be this really dangerous drug, right? So we thought, the press is going to be all over us, and you're going to have to keep people overnight. Well, what about mescaline? We know the Native American Church had used that, and but again, it's 8 to 10 hours, and a lot of people that take mescaline get nauseous or even vomit. So you don't want to take people that are dying and make them sicker. So, well, what else do we have? Well, there's psilocybin that had been used in South America for thousands of years. It lasts 4 to 6 hours. So you could bring people in the morning, give it to them, and then discharge them at the end of the day, have somebody drive them home. So we thought that made the most sense. And then I was working on improving the synthesis of psilocybin. So that's basically it. It's a short acting, it was orally active, and we didn't think anybody knew what it was. When I got on a plane to fly back then, I'd tell somebody that we were doing work with psilocybin, they'd go, what's psilocybin? I never heard of that. And I'd say, well, have you ever heard of magic mushrooms? Oh, yeah, we took those back in college. I said, well, psilocybin is the active ingredient in magic mushrooms. And so that's how far it's gone. I can say psilocybin today and everybody knows what I'm talking about. Well, everybody in this talk probably. But yeah, so it was really, we chose it for shortness of action, recognition of its safety because it's been used for so long. And it didn't make people sick.
Thank you so much for the answer.
Yannick, if you'd like to give your question, please.
Yeah, thank you for a very nice presentation, Dr. David. I was a bit curious about the rat experiment when you trained them to press the panel on the right side, and that was LSD. Have you tried to say if they press the panel for like a month or something like that, and they're getting used to the right panel is the LSD, then switch it? So the left panel was the LSD now, and the right was the placebo?
That would be cruel. So they're pressing this lever to get a rat candy, right? So they're used to pressing it and getting a rat candy. So in the beginning, when you first start training them, they move back and forth. In the beginning, you put them in a cage with only one lever. And so rats don't know, in the wild, they don't press the lever. So the rat will explore and he'll just wander around, you'll see him, and he'll stand on the lever and sniff at the top. And when he stands on it, the thing that delivers the food pellet makes a loud click. And he comes down and he starts looking around and he sticks his nose in that hole and he finds out there's a rat candy in there. So that doesn't mean anything. But about the third or fourth or fifth time he does that and he gets a rat candy, he starts saying, somehow when I'm over next to this lever, I'm getting a reward. So he learns to press a single lever. And then we go to the cage, we put 2 levers in. Well, now what's he do? So now he still goes around, but he finds out, depending on what we gave him, that 1 lever works. Now, I don't know how long it would take to extinguish that, but I'm sure you probably could do it because they've learned to press a lever. So what's happened sometimes when we've forgotten to turn the correct lever on, we put them in, they give them a drug, and they'll press and nothing happens, and they'll press and nothing happens, and they just give up. they don't go and try the other lever. But I suppose you probably could find a way to get them to switch them, but you'd have to extinguish their first behavior and get them to press on the other lever. I mean, it would be possible, but remember, it takes 2 to 3 months to train one of these rats. And if you switch it to another side, it's going to probably take another 2 or 3 months to extinguish the previous behavior and go with another behavior. And people aren't using rats so much anymore, because when I started my research, rats were maybe $2, $2.50 US. And now they're like $50 or $60 a piece. And people are using mice because now mice are $2 or $3 or $4. They're cheaper. And mice aren't nearly as good a model as rats are, but they're so much cheaper and easier to maintain. But we always did our work with rats.
Okay, thank you so much for that answer.
Thank you. Rai, did you have another question, or does anyone else have a question I should ask? Just to...
Alex, there's a couple of questions in the chat if you want to read.
Oh,
those
there
out.
are. Thanks for letting me know. I completely missed that. Ronald, would you like to ask your question just vocally, or I can read it out if that's more comfortable?
Sure. Can you hear me okay?
Yep.
Yeah.
I can hear you fine.
I was interested, Dr. Nichols, in the access to the formula for synthesis of psilocybin. I had read something about it being in the public domain and involvement with the USONA Institute, and then I guess some tension perhaps with Compass pathways. And do you have some information on that you could share?
I have a lot of information on that. When we decided, I worked with Strassman, and then Rick sent in a grant. He wanted to use psilocybin, and there was no source of psilocybin. And so we were going to make it. And actually, Rick Strassman got really pissed off at me because we didn't get it made in time. He wanted to do the experiment. And the way that Albert Hofmann had originally made psilocybin was by using a reagent called dibenzyl phosphoryl chloride. And I didn't know anything about it, but I gave my technician, Stuart, I said, well, here's the patent to use. Let's follow that. Stuart came back in and said, this reagent, this dibenzyl phosphoryl chloride, he said, it can explode. If you have it as a neat solvent, it'll explode. And he says, I don't want to work with it. And so, of course, I said, oh, no, if it's going to, if it's that dangerous, we don't want to work with it. So we spent a long time. He tried to, because the last step, you can make psilocin, but putting the phosphate on has been the hassle. So we tried a bunch of different reagents. The first batch of psilocybin we made, we used a pentafluorophenyl dichlorophosphate, and it worked okay, but we had trouble getting the byproduct out, which is a pentafluorophenol. It wanted to stick to the psilocin really tough, and we had a lot of trouble getting it. So we had to find another way to do it. Finally, Stuart went back and looked at the original patent that Hoffman had filed, and he said you can put the phosphate on with the dibenzyl phosphoryl chloride, or you can use tetrabenzyl pyrophosphate. And I hadn't heard about that, so Stuart said, let's do that. So that was a crystalline white material that was completely safe. And so then we started using that. So when Compass started up, he originally was going to work in the Isle of Man, and they changed whoever runs that place. I don't know anything about British politics. And the person who took over as whoever was running the Isle of Man wouldn't let him do the study. He was going to do it in a hospice type situation, as I recall. So then he decided he was going to, George Goldsmith and Katya, his wife, they were going to start their own company to treat it. Their son had been, I think, severely depressed and had taken psychedelic mushrooms and they had gone into remission. So they thought, well, this is really important stuff. So he asked me if I would help consult on that. And I said, yeah. So I consulted pro bono on their synthesis. They made it the same way we made it. Then Bill Linton he started USONA, and they wanted to do a study, and he wanted to get some psilocybin from Compass. Compass had originally gone to Bill Linton and said, do you want to collaborate on the cost? Because the things that I made were like $10,000 for 10 grams. But if you make GMP, it's like it's super expensive, like $500,000. So he went to Bill Linton, but Bill Linton, who's the president of Promega and is the founder of USONA, he didn't like George Goldsmith for whatever reason. And he didn't want to collaborate with him. He said, no, you do your own thing, we're gonna do our own thing. So he hired, USONA hired a company named Sterling. And on the advice of Rick Doblin, who had gotten Sterling to make his GMP MDMA. And Sterling screwed up the synthesis really badly. And so Bill Linton then went to Compass, who by then had made a kilo or so of psilocybin, GMP psilocybin, and wanted to get it. And George Goldsmith says, well, you can have it, but your safety data has to go toward our application to the IND and the European Medicines Agency. And so Bill Linton said, no, I'm not going to do that. I'm not going to give you the data. I'm going to find another place. So he searched around and found another place. I still don't know who he found, but they made quite a bit of psilocybin for him. And then one of his chemists, Robert Cargbo, who was a process chemist at Lilly before he went to work for USONA, decided to try to put the phosphate on just with a reagent called phosphorus oxychloride, which is commercially available, and it worked. So then USONA used that procedure to make, I think, 1.3 kilograms of GMP psilocybin. Now that method has been used by other people, but it became a personal vendetta because one of the board members of USONA is Kerry Turnbull, who is also a board member of Hefter. And Kerry was going to fund this study that they couldn't get the psilocybin for, and he said he lost $1 million. So then he got really mad at George and Katya, and so they started this open science thing to try to break the patent. And it's kind of really sad. I hate to see it that these people are supposed to be enlightened or attacking each other, but yeah, that's the general outline of the details.
Great, thank you. Jesper, you have a question? Is he here? I just think it's a lot nicer for him to physically ask it rather than leave it as text. I don't know if he's still here.
Unmute.
Oh, there you are.
Sorry. Thank you, David, for an excellent talk. It's been a few years since I saw you at a PhD defense, if you remember, in a restaurant afterwards.
Oh yeah, who's PhD?
With Jesper Christensen, it was Sebastian.
Oh, Sebastian.
PhD defense.
But Sebastian left the field, didn't he?
He did, sadly.
Yeah.
We got the grant together, and then he left right after we got the grant, actually. That was sad.
Yeah,
Anyway, I have a question. I have a question to you, David, about the drug discrimination, because we've had a discussion about what it really shows. The rat can tell us that this is an LSD-like experience or a psilocybin-like experience or whatever. But does that tell us anything? What is your take on how much does it tell us about the psychedelic experience? Or does it just tell us if this is a 5-HT2A receptor agonist experience? So my question is, have you tested Lisuride, which, as far as I'm aware, is a 5-HT2A receptor agonist that does not elicit a psychedelic experience? So do you know if that has been tested? Have you?
There was a student, he's passed away, unfortunately, that I forget who he worked for now, who looked at discriminating Lisuride and LSD and were able to train the rats to tell the difference. Lisuride has low efficacy at the 2A receptor and actually has powerful dopaminergic effects. So drug discrimination picks up the most salient feature to the rats. Well, in humans, Lisuride is not hallucinogenic either. We've got some BOL being tested, and people are starting to look at 2-bromo-LSD BOL, which was used as a placebo, but actually it activates the serotonin 2A receptor too. So there's an issue of functional selectivity. What are the signaling pathways? And it's I haven't seen the data for Lisuride. Brian Roth, I know, is doing Lisuride in his he's doing he has about a library of 50 different hallucinogens plus some non-hallucinogens, and they've got BOL and Lisuride both in there. My understanding though is that glyceride is a very low efficacy, even weaker than LSD. LSD at the phosphoinositide turnover is about 30% efficacy. And I don't know, but it's more potent in arrestin. It's a full agonist in arrestin. And I don't know what, I haven't seen the data for BOL. It may well be that BOL is not activating arrestin. So the idea of functional selectivity is going to be important, because if it requires arrestin, I've seen data, for example, in arrestin knockout mice, arrestin 2, not arrestin 1, but in arrestin 2 knockout mice, they don't exhibit the behavior that wild mice do when they're given LSD. So it may be that arrestin is
that the head twitch response, that they don't do the head twitch?
He's looking at open field, a bunch of open field behaviors, but not, I haven't seen, I can't recall. He probably has done head twitch, but I think he doesn't see that either. So it may be that it's functional selectivity, in addition to the fact that glyceride is a weak partial agonist. But you'll see those data probably published, maybe that paper, I'm guessing that paper is almost ready to submit. I'm guessing by the end of the year, you'll probably see something about that, where you'll see the most complete set of data that you've ever seen on any of these, because we get mescaline analogs, tryptamine analogs, LSD analogs, FLAD, LSD, ALAD, and they're being put in all of Brian Ross receptor screens, looking at all the receptors they activate, and including arrestin. So, but it's more likely something related to functional selectivity than anything else. That's the one people always raise. Well, glyceride, that's an agonist, isn't it? And then we just started looking at BOL, because that was for years. When I first got a sample for Brian, he said, "Oh, we tested that, it's inactive." And then I got a new sample that had been recently made, and they started testing. I said, "Oh, this has got activity." So it's a really interesting question. It bears on the ultimate mechanism of action. So I think we'll see more of that this year later on. It's a work in progress.
Very interesting answer. Thank you, David.
I'm not very good at chemistry at all. Is BOL related to BOD, or did they just happen to have similar naming formats?
BOL, I think Sandoz called it BOL 148 or something like that.
Right. Okay. I'll have to look into it more.
And his LSD 25, but then he had other compounds, and they all just had names he made up for him. So
right, fair enough. Okay. There's no particular relation there. Great. Okay. Rye, did you have one more question? Just so I make sure I've got everyone, if there's still time for that.
Yeah. So it was a question that I've had in my mind before this talk, but especially like I was reminded of it when you showed the diagram of the magnets, the MEG diagram of the brain, and I was wondering, from a pharmacologist's perspective, what do you think has the biggest room for improvement in terms of measuring equipment that you use within the lab? Because I know that for MEG, in particular, they're starting to design these helmets that can be worn. So there's a larger range of experiments that can be done just because the patient can move. I was wondering if there's a sort of similar analog to that in pharmacology that you could if you could theorize would be of use.
I think we have, Franz Vollenweider has done a lot of EEG work. Then you've got fMRI to look at functional connectivity. Magnetoencephalography is interesting to me, but those machines are pretty expensive. But I think they're done with a squid helmet, right? Superconducting quantum interference detector, something like that. And those can get deeper levels. But I think if MEG was really developed a little bit more, and I don't know anything about the software for these. I mean, it's unbelievable what the software takes to analyze these data, and the amount of data you have to store to analyze. But I think, until probably 10 years ago, I don't think we even had the tools to do the stuff we're doing now. I mean, just if you look at the computational power now, my son's just got a $7,000 deal for some kind of a computer that's got 25 cores. What, for $7,000? I mean, you couldn't have done that 10 years ago. And I know Brian's got a guy that's doing his virtual docking that's got 1,000 cores. So I think the power of the software is making things possible that we couldn't have conceived before. And EEG is more surface. MEG is, I guess, goes a little deeper. fMRI, of course, is looking at oxygen isotopes, oxygen in there. But I think we more or less have the tools. They maybe need to be refined a little bit, but I think as far as the brain and doing scanning, unless there's some new breakthrough. I got a reminder of my meeting at 3:30. Yeah, I think the tools are probably fine. I had a dream when I was a graduate student. I was in this auditorium and somebody was sitting under a big thing on their head. And years later, I realized it was a magnetoencephalography. It was a squid and superconducting head and everything. They didn't have those at the time that I was dreaming this. So it's kind of weird. So I always wanted to see somebody using MEG. And then, of course, a paper came out of David Nutt and Cara Harris using MEG. They don't use it much. And I don't know what the limitations are. I suspect part of it's getting time on it. And they're pretty expensive.
You also have to keep the patient very, very still.
Yeah.
And they are expensive because because it's a superconducting detector. You have to keep it consistently cooled on, usually with liquid nitrogen. So I can't. It must have cost at least in the hundreds of thousands, if not millions, to just run these.
Yeah, I know they use them for, I think, a lot for diagnosing epilepsy. And there's a few centers, like Cincinnati, University of Cincinnati bought one, said, oh, we bought us an MEG and we can do this. We're the only medical center around that can do it. So I suspect they're rare, but I think that's an interesting technology that I'd like to see more of used for psychedelics. But I don't know what's going to happen in the UK. The US, they're having now, they had a panel today that goes tomorrow that was sponsored by NIDA. They had another panel that was sponsored by Proceedings of the National Academy of Sciences. There's another panel coming up in May that I think is also sponsored by NIH. And today, one of the comments was made like, well, what do you need to see happen? And Brian Roth was one of the panelists. He says, we need this work to be funded. They're spending money on all kinds of things, and there's no money really going into understanding these. And it looks like they are the new breakthrough for psychiatry. And there should be more research finding out how they work and what the mechanism is. So sooner or later, this is going to happen. And then once there's money, then people can go in with the center and say, we've got 20 people and we want to do MEG, and we've got a match from some philanthropist and, some buying some of those pieces of equipment for this.
Well, thanks for your answer.
I wonder briefly as well about kind of potentials in the future for lesser known substances for therapeutic value. I mean, I'm reminded of, there was, I think it was in the 70s, I can't remember which trial it was, but using DPT in palliative care for near-death cancer patients. And that to me is super interesting. And I guess it's enough of a difficulty kind of trying to find research with sort of classical psychedelics. But I wonder if there's anything like that or there could be anything like that on the horizon for sort of approaches with lesser known substances, because I'm personally of the kind of opinion that a lot of these ones might have value even if they fall out of the traditional remit of substance use. I don't know, maybe I'm rambling now.
I think, drug discovery is really expensive. The study that we sponsored at New York University, which was done at a university with low overhead, et cetera, that was 96 subjects. And I think Hefter Institute rounded up around about $3 million to fund that study.
Wow. Yeah.
That's relatively inexpensive. You're talking about tens of millions of dollars for a study. I saw one of these companies one of these startups for psychedelics. They were going to develop 2CB for women's health. I know a little bit about 2CB, and I'm going, 2CB for women's health? Like, what?
I mean,
so talking about the cost of things. So I made about 1.5 kilos of high purity MDMA for Rick Doblin for all of his phase 1 and 2 studies. He got 1 kilo of GMP material made. It cost $400,000.
Yeah.
So if it had only been available like that in the beginning, none of this field would have gone off. And similarly, Rick Strassman, he paid me a few $1,000s, and Johns Hopkins, I made 20 grams of psilocybin for $10,000 and stuff like that. Just cost of materials and graduate students.
Yeah.
If you're actually going to start a program, so suppose you need to buy a kilo of 2CB, which is fairly easy to make. It's still going to cost you. They'll do process. The company that does it will want to do process research and optimize the process. So you'll pay for all that. And suppose when all is said and done, they make a kilo of 2CB for you for, say it's $400,000. Then you're going to have to get toxicology, preclinical toxicology in 2 species. That's going to cost you a couple $100,000. We've been lucky because LSD, psilocybin, and some of these others that are being tested have been used enough that the FDA is not real worried. But if you actually want to make it into a drug, like you're going to still have to do formal toxicology studies.
Right. So
you think, okay, you're going to make 2CB into a drug. You're looking at an upfront cost that's going to be, $400,000, $500,000, $600,000, $700,000, and you don't really have a clue what you're going to use it for. How do you get investors to come up with that? And that's not a lot of money. I know deals where investors are closing on 20 million, 30 million, 40 million, et cetera. You're not going to get investors to come up with that kind of money for an unknown compound.
No, cool.
Now, I came up with a compound called 6-APB. And I've gotten 3 emails from people saying, did you make that? Are you the one that made that? And I actually didn't. I made 6-AP dihydrobenzofuran, but 6-APB apparently is a research chemical that's available for sale. 3 people have told me, one of them said he had severe ADD or ADHD. And then another woman said, like I was so depressed and if I took this, my whole life changed. I'm going, this is pretty interesting. So if you get a signal like that and you had a company with a lot of money, you could say, let's make a 100-gram batch of this, not GMP. Let's do some animal studies and see, is there something to this? And you'd have a basis to start, but somebody's telling you that. If you look at PCAL, and I made a bunch of the molecules that are actually in there, but I couldn't do human studies, so I'd send them to Sasha. But if you look at PCAL and you look through there, you'd have to find something that looked like, oh, this would be really good for something. And what do we have at? We have MDMA. And actually that was, that again is another one of these things that was discovered on the recreational, market. So I don't know. I mean, conceivably, there are some things in there that Sasha has made, or either tryptamines or phenethylamines. Conceivably, there would be things that would have some advantages. If we find out what the actual underlying events are, the pharmacology that leads to antidepressant activity. They talk about a synaptogenesis and spinogenesis. You can run some of those in vitro. So it's conceivable you could get somebody to go through and make, 100 of the compounds from PCAL that looked like they were non-toxic, put them in an in vitro assay and see whether any of them, had an effect. But so far we don't have the in vitro screens to know what to look for.
Right.
So that would be, I think, the first thing, if we got good in vitro screens for some particular thing where we know what's wrong, then we can do an in vitro screen. Then you could follow up. But just to pick one and say, like this company in Canada did, we're develop 2CB for some women's health issue. And I heard that, I thought, what? So
yeah, no, fair enough. Yeah, I suppose I'm always interested in reading about the effects of these new ones, and just that's a lot of where my perspective comes from, is that psychonautic sort of. But yeah, that makes complete sense.
And I've made compounds that in rat studies look like they would be interested. I've never been able to get anybody interested. I don't know what they would be good for, but non very specific serotonin releasing agents, and I made an MDMA analog, which looked like it had the same effects as MDMA, but didn't produce the euphoria.
Was this
So-
MDMAI? No, I'm only thinking about
MBDB.
Oh, okay, yeah.
It
yeah.
never caught on because it didn't produce euphoria.
Oh, that's not going to work well in the commercial market, is it?
Yeah, but you know, once you have a company that has some money, you can say, look, we should do some small studies with animals because we've got cash to burn, etc. It takes some investment, some money. You can't just say, oh, eeny, meeny, miny, mo, let's pick this one out of page 35 of P. Collin.
Yeah.
We're going to develop it into something.
Yeah, no, fair enough. That makes a lot of sense. Okay.
I have a Zoom thing right now, so I'm afraid I'm going to have to leave. I'd love to talk some more, but
thank you so much for your time. This has been really appreciated. All right. Thank you very much.
All right. Have a good day or evening, as the case may be.
Cheers. Have a good day.